Dr. Noam Sobel, a professor of neurobiology in the Department of Brain Sciences at the Weizmann Institute of Science, is the guest on this episode of Huberman Lab Podcast. Dr. Huberman introduces Dr. Sobel's laboratory studies on olfaction and chemosensation, or the sense of smell and the ability to respond to chemicals in the environment. The discussion begins with the fact that humans have the ability to smell things around them, as well as dogs. Dr. Sobel asserts that humans are incredibly good at sensing the chemical world around them, as they are constantly taking chemicals from other people around them when they are nearby. And they process this information to determine many things about them, such as how stressed they are or their hormone levels. These chemical interactions have an impact on emotions, decision-making, and the relationships that individuals choose to form. In addition to interactions with others, humans are also sensing their own odors, even though they may not notice their own smell. Dr. Sobel's laboratory discovered that humans periodically smell themselves deliberately throughout the day to change their cognition and behavior. Furthermore, he notes that tears from others can impact an individual's hormone levels in powerful ways. Dr. Huberman recounts a peculiar experience observing an experiment being conducted by Dr. Sobel's laboratory at UC Berkeley, where individuals were following a scent trail that led to a scent buried underground with a high degree of fidelity. This experiment led to other important discoveries related to human olfaction and chemosensation. Dr. Sobel explains that our ability to smell involves the nose and mouth, with retronasal olfaction being a big part of the contribution of olfaction to food and taste. Airborne molecules travel up our nose and interact with the olfactory epithelium, a sensory surface with about 6 or 7 million receptors in humans. The system is organized into different types of receptors that are specialized in detecting certain types of odorants. Dr. Sobel then explains a funny anecdote looking into the origin of a claim that there are 1 billion receptors in the bloodhound's sense of smell. They found out that the origin of the claim was completely made up and shed light on how much of what is in scientific and medical textbooks can be wrong. They then moved on to discuss the neural signal that odorants create when interacting with receptors in our epithelium, which then travels via the olfactory nerve to the olfactory bulb in the brain. Dr. Sobel explained that losing the sense of smell due to trauma is because of a contre-coup injury, which generates a shearing motion on the cribriform plate, severing the olfactory nerve. If the olfactory nerve is completely severed, it is lost forever, as it is among the few central nervous system neurons in adult humans that cannot regenerate. However, partial nerve damage or something showing up after a short while may still have a good chance of recovery. Dr. Sobel also mentioned that if a person does not get their sense of smell back within a year to a year and a half, they may never get it back. Dr. Sobel mentions that alpha-lipoic acid can accelerate the recovery of smell. However, he notes that other chemosensory sensitive nerves also contribute to our sense of smell. For example, the trigeminal nerve, which causes the sensation of burning when cutting onions. Dr. Sobel explains that smelling things is important for maintaining olfactory neuron connections, as the olfactory system is a use-it-or-lose-it system. He cites studies that showed that olfactory training can help recover smell, which involves smelling a variety of different scents intentionally and constantly. Additionally, Dr. Sobel highlights a 2014 study in which researchers assessed the levels of C14 in postmortem brains to determine the extent of olfactory neuron regeneration in humans, a topic that is still debated among scientists. Dr. Noam Sobel explains that in the olfactory system, the receptors of one subtype converge to 1 or 2 locations in the olfactory bulb called glomeruli. Each receptor subtype recognizes a small subset of odotopes, molecular shapes of odor molecules, and each odotope activates a different subset of receptors. This potentially creates an insane combinatorial space of potentially 350 dimensions in humans. However, due to convergence, each odor activates specific glomeruli that reflect the receptor identity, creating a map of odor on the olfactory bulb. Information then goes from the olfactory bulb to several targets in the brain, including the primary olfactory cortex or piriform cortex, rhinocortex, amygdala, hypothalamus, and cerebellum. The understanding of how coding occurs in the rest of the brain is murky, but the memories of odors are thought to be more robust than the memories of other perceptual events in our lives. Dr. Sobel explains that the first time we experience a smell generates a particularly robust representation more than other sensory stimuli. This effect is called the privileged representation of early olfactory associations. There is a lot of research on this topic that suggests there is something special about olfaction, although there might also be something special about vision or audition. Dr. Sobel jokes that he tries to explain everything through the olfactory system, and for him, everything is olfactory. Sobel explains how people can improve their sense of smell, as humans already have a remarkable sense of smell. For instance, humans can detect mercaptans, an additive to cooking gas, at the threshold level of 0.2 parts per billion, which machines cannot detect effectively. Sobel also shares an experiment he ran with his colleagues during a lab picnic at UC Berkeley, where they talked about humans' ability to track odor like a dog. They conducted an experiment, which Sobel has a video of, demonstrating that humans could track odor effectively. Dr. Sobel describes the experiment he conducted, where participants were blindfolded and had all other senses blocked off except for their sense of smell. They were then asked to follow a chocolate trail that Dr. Sobel had created in the grass using odor-impregnated twine buried under the surface. Participants were also wearing a sensor pack to measure their nasal airflow in real time and track their behavior using RTK GPS. Dr. Sobel found that participants were able to successfully track the odor trail even without prior training, and after an average of 4 days of training, the main limiting factor was the speed at which they could crawl. He also discovered that having 2 separate nostrils contributed to better tracking performance, as opposed to combining both nostrils into one centralized nostril. This is because the human olfactory system is highly lateralized, meaning that the representation of smells in the brain is on both sides of the brain, just like our 2 eyes. Having 2 separate nostrils allows us to gain depth perception information, perceive motion better, and locate sounds more accurately. Regarding the mechanics of sniffing, Dr. Sobel explains that they did measure and record sniffing behavior during the experiment, although it was not a major component of their analysis. He notes that sniffing behavior is a huge portion of their work in the lab, and that they are currently doing more research on the topic. Dr. Noam Sobel talks about an experiment that showcases the nasal cycle, which is often overlooked in neuroscience. He suggests occluding one nostril and then the other and noticing the difference in airflow. This is a reflection of the nasal cycle, where the high-flow nostril and low-flow nostril alternate every 2.5 hours on average in an absolute way. The shift from left to right nostril is linked to balance in the autonomic nervous system. The autonomic nervous system has a sympathetic and parasympathetic component, which are in balance or imbalance in many diseases. The seesaw of alertness and calm is governed by the interplay between the sympathetic and parasympathetic nervous systems. The nasal cycle is a marker on balance in the autonomic nervous system, but it is not currently being used. To address this issue, Dr. Sobel's team has built a wearable device called the nasal halter, which is pasted to the body, measures airflow in each nostril separately, and logs it for 24 hours. They're collecting these 24-hour recordings to measure it as a disease marker. Using the nasal halter, they have identified nasal airflow differences between ADHD and non-ADHD adults and can determine if adults are on Ritalin or not. However, Dr. Sobel admits that he does not have a clear answer on whether airflow through one nostril is reflective of sympathetic nervous system dominance, or if the alternating left-right nostril periodicity is simply reflective of an overall balancing of the seesaw. While the yogis will be interested in this mechanism, Dr. Sobel's team wants to gain control of it and help people who might benefit from it. Dr. Noam Sobel talks about an experiment they conducted with yoga practitioners to test their ability to control the flow of air through their nostrils. Out of the 14 yoga teachers they tested, none of them were able to willfully switch between left and right nostril flow without physically manipulating their nostrils. This led to a discussion about the underlying cause of the alternating nasal cycle and whether it reflects a shift in the autonomic nervous system or if it is the cause of the shift. They further discuss the architecture of the smelling system with converging olfactory receptors in the glomeruli and a mirror representation on both sides of the brain. However, Dr. Sobel doesn't believe that the nasal cycle is related to olfactory processing. He suggests that the alternating nasal flow is a different story about brain function, with potentially important implications for autonomic arousal. They acknowledge the need to dispel myths about the right brain-left brain dichotomy and focus on the science behind this phenomenon. Dr. Noam Sobel is discussing the sniffing brain approach, where nasal inhalation drives aspects of neural activity and cognitive processing. This approach is inspired by the fact that the mammalian brain evolved from olfaction, which depends on sniffing. The brain processes information on inhalation onset. Dr. Sobel conducted a study on visual spatial processing and found that people performed significantly better on inhalation versus exhalation. Dr. Sobel recommends nasal breathing for health reasons as well as cognitive benefits, as other labs are finding the same results. The olfactory sensory neurons are located at the tops of nostrils and are the only place where the brain meets the outside world directly. Loss of the sense of smell is one of the earliest signs of neurodegenerative diseases, such as Parkinson's and Alzheimer's. Alzheimer's may be the result of a pathogen entering the brain through the olfactory system. Non-invasive readouts of potential neurodegeneration, such as visual tests, may also be useful as the retinas are part of the brain. Dr. Sobel explains the idea of using olfactory tests to track changes over time in an individual's sense of smell. This could potentially reveal if changes in an individual's sense of smell could be a sign of Alzheimer's or other diseases. However, the reason this hasn't been done yet is because olfaction has not been effectively digitized. Standard clinical tests, such as the University of Pennsylvania Smell Identification Test, UPSIT, and Sniff and Sticks, do exist, but are not as convenient as vision or audition tests. Dr. Sobel explains that olfaction has poor control of the stimulus, making it difficult to generate precise information. Additionally, there is congenital anosmia, where a person is born without a sense of smell. It is diagnosed on average at the age of 14, which can cause social and life-changing problems. Dr. Sobel notes that people with congenital anosmia don't have nostrils, although there is a rare condition where children are born without them. Dr. Sobel states that most people who are born with anosmia do not have olfactory bulbs in adulthood. However, some people have remnants of the olfactory bulbs. Dr. Huberman asks if their endocrine system is affected by anosmia, to which Dr. Sobel responds that there is a direct link between the two. Some people with anosmia have conditions like Kallmann syndrome, which leads to hypogonadism in men, affecting testosterone hormone production. Dr. Sobel further explains that olfactory systems are linked to the hypothalamic systems that regulate the release of gonadotropins, controlling hormone production. While not all congenital anosmic individuals have Kallmann syndrome, almost all people who have Kallmann syndrome are anosmic. The possibility of a female equivalent of Kallmann syndrome being associated with anosmia is also raised. This discussion leads to Dr. Huberman segueing to talk about his ability to identify his friends by the scent of their clothes during his childhood. Dr. Noam Sobel then explains how humans communicate subconsciously through chemical signals. He mentions that humans process this information subconsciously and that there are many studies in his lab dedicated to studying this phenomenon. One amusing example he provides is that dogs sniff each other upon meeting, but humans do not. However, he suggests that humans do indeed sniff each other subconsciously through various mechanisms, such as handshaking. Dr. Sobel's lab conducted research on handshaking and found that people touch their nose, forehead, or chin after shaking someone's hand. To prove this, they conducted an experiment where they brought in participants and videotaped them without their knowledge. During the experiment, they observed how much the participants touched their face and then later watched the videos to quantify this behavior. They then introduced an experimenter who either shook the participant's hand or not and observed how often participants touched their faces after the handshake. In the study, participants shook hands with an experimenter who emitted either a pleasant or unpleasant odor from a watch. The researchers found that participants more frequently brought the hand that had shaken with the pleasant-smelling experimenter to their noses than the unpleasant-smelling experimenter. Dr. Sobel explains that people not only smelled the hand that shook, but also the other hand, which led the researchers to hypothesize that there might be something going on related to comparing self to other in the olfactory behavior. The conversation then shifts to a study conducted by Dr. Sobel's lab on clique friendships, or friendships that form rapidly because of a sense of immediate connection. Dr. Sobel discusses how this may be related to similarities in body odor. The researchers recruited clique friends and tested whether they were more similar in their body odor than non-clique friends. They found that click friends were indeed more similar to each other in their body odor and went on to have other participants smell click friends versus non-click friends to confirm the finding. To test the relationship between similarity in smell and social interaction, Sobel conducted an experiment where total strangers smelled each other with an electronic nose before participating in a nonverbal interaction called the mirror game. Interestingly, although Sobel's prediction that people who smell similar to each other would be better at the mirror game did not pan out, the electronic nose was still able to predict how much each participant wanted to be friends with every other participant. Sobel argues that this experiment demonstrates a causal relationship between smell and friendship, and that humans are constantly smelling themselves and others in their daily lives. In addition, Sobel explains that the immune composition of an individual's smell is also important in choosing a romantic partner, as animals and humans tend to choose individuals whose immune composition is most different from their own. Sobel notes that there is extensive research on the topic, and that there is evidence to support this mechanism in humans as well. Dr. Sobel explains the subsystems in our nose that transduce odorants, which is primarily the olfactory system and the trigeminal nerve. Additionally, most terrestrial mammals have a vulnerable nasal organ, VNO, which is a small pit in the nasal passage connected to the accessory olfactory bulb. This subsystem is responsible for processing pheromonal effects, or odorants that are emitted to influence behavior or hormonal state. The most remarkable of these effects is the Bruce effect, which causes pregnant mice to abort their pregnancy when exposed to the scent of a non-stud male during early critical stages of pregnancy. This effect is dependent on the VNO, as ablating it eliminates the effect. Although humans are believed to lack a functional VNO, it is important to note that pheromonal effects can still occur through the manufactory system. Dr. Noam Sobel brings up the psychological and physiological changes that occur while females exhibit spontaneous abortions or miscarriages after coming into contact with an unknown male. Although it's been widely studied in mice, Dr. Sobel is not completely convinced that it doesn't exist in humans. Humans have an enormous ratio of spontaneous miscarriages, most of which occur in the first trimester. While some studies suggest that 90% of all pregnancies end in miscarriage, more conservative estimates place it at 50%. They found that these women were double and sometimes more than double the performance level as compared to others who have not experienced pregnancy loss. They conducted control studies and found that these women were not better at detecting other odors besides their spouse's body odor. They also used fMRI to measure the brain response of women experiencing pregnancy loss to stranger male body odor. They found that there was a huge difference in the hippocampus between these women and the control group. Dr. Sobel and Dr. Huberman also discussed the term pheromones and how it is a problematic term. They found that regardless of the answer to whether humans have pheromone effects or not, humans emit chemical signals that influence each other's physiology and behavior. They talked about how the term pheromone was put forth to describe insect behavior and was later applied to animals. However, human chemosignals influence each other's behavior in several ways, and humans emit a particular body odor when they are in a state of fear. They found that the smell of fear is contagious and can lead to an increase in the autonomic arousal of other humans. Dr. Sobel then talks about the phenomenon of romantic partners leaving articles of clothing at each other's homes in order to bring about positive connotations of the other partner. He also raises questions about whether or not the mourning period after a breakup has something to do with an olfactory unlearning process. Dr. Sobel emphasizes that humans are incredibly olfactory animals, and even not in the nuances, our sense of smell is of great importance. He gives examples to stress that the sense of smell affects the two most basic behaviors that sustain us, eating and choosing a mate. Sobel refers to a molecular compound called hexadecanal that promotes social buffering in mice. This chemosignal is highly conserved among mammals, and its presence promotes a feel-good-together vibe in mice. Sobel and his colleagues received hexadecanal from their friend and colleague, Heinz Breer, who suggested they study the molecule's effects on humans. Sobel's student, Olva Mishor, designed a paradigm to study human aggression using hexadecanal. They used the Tyler aggression paradigm, where participants were paired with a jerk computer algorithm who offered them very little of the money being divided in each round. Participants played 2 games against their opponent. the second one being a reaction time game where the winner could also elect to blast their opponent with a loud sound. Sobel and his team found that participants exposed to hexadecanal were less likely to react with aggression, supporting the idea that the molecule promotes social bonding across many species. The results also showed that hexadecanal consistently reduced aggression in men, but increased aggression in women. Sobel was surprised by this gender difference and doubted the results, so he replicated the experiment within an fMRI magnet to track brain activity. The fMRI results showed that hexadecanal increased activity in the left angular gyrus, an area involved in social appraisal. However, this activation was the same in both men and women, even though their behavior went in opposite directions. Sobel then performed a follow-up analysis on functional connectivity and found that hexadecanal increased connectivity in men and decreased it in women, suggesting that the default brain function for aggression is present in both men and women, but hexadecanal modulates its control differently between the sexes. Dr. Noam Sobel talks about the results of a recently published paper from Japan that looked at the odors emanating from baby heads, specifically if hexadecanal was one of the molecules reported in the paper. Hexadecanal is a semi-volatile that can make mothers more aggressive and fathers less aggressive towards their offspring, making this beneficial for the babies. The scientists found that hexadecanal is the most abundant semi-volatile in baby heads, suggesting that babies conduct chemical warfare to control the aggression of the adults around them for their own benefit. The scientists then discussed the coordination of menstrual cycles among co-housed women or friends, a phenomenon first discovered by Barbara McClintock in a 1971 Nature paper. While many women claim that their cycles become coordinated when spending time with their friends, subsequent research has called this into question. McClintock later published a follow-up paper in 1998 where they collected sweat from donor women and deposited it on the upper lip of recipient women, suggesting that there could be an olfactory or chemosensing aspect to the synchronization of menstrual cycles. The study found that sweat from the follicular phase extended recipients' cycles, while sweat from the ovulatory phase shortened them. However, the study's findings have since been questioned due to the tricky statistics of cyclic events. Dr. Huberman notes that the study's potential replication is currently a topic of interest in his lab. He also emphasizes the significance of olfactory effects in humans, noting that they're linked to primal mechanisms in the brain and may have real-world applications. They note that the use of sweat in human social chemosignaling research is common among labs worldwide, with most only studying sweat from above the waistline. They then discuss the study of bodily fluids for their chemical signaling properties, which can be used to develop treatments for conditions such as anxiety. The particular focus is on tears as a potential medium for social chemosignaling, as emotions in situations where nonverbal communication is critical and there is a need for chemical signals. Tears seem to serve no function beyond ocular maintenance, and Charles Darwin, in his book, The Showing of the Emotions in Men and Animals, could not find a functional antecedent for tears. So the purpose of the fluid remains perplexing. With this in mind, Dr. Sobel and his team harvested emotional tears for chemical analysis, and significant gender bias was observed. Dr. Sobel mentions the study where participants were asked if they cry easily and to name a specific movie scene that makes them cry. The researchers collected a full ml of tears from effective criers in about 15 minutes and discovered that these tears were completely odorless. When men sniffed tears collected from women, even without perceiving any odor, they experienced a significant reduction in free testosterone within 30 minutes. Dr. Sobel notes that the effect is a strong one and that an independent group in South Korea was able to replicate it. However, the researchers note that only a few participants could consistently cry on demand, indicating that cultural norms and learned behavior play a large role in the ability to cry easily. Additionally, brain activity was lowered under an arousing state in both the hypothalamus and the fusiform gyrus. Researchers also found that sniffing tears might lower aggression in men, as demonstrated by the replication of the experiment involving the titration aggression paradigm. Though the study was significant, the weaker effect on behavior led to criticisms of the research by a lab in Holland, led by a researcher named Vingerhots. The lab claimed that emotional tears were uniquely human and did not like the findings. However, the tear study showed that tears are a chemosignaling mechanism like all animals. Furthermore, a paper in Current Biology confirmed that dogs emit emotional tears when they reunite with their owners, which can influence oxytocin levels in both the dog and the human. Dr. Sobel believes that dogs tap into the same circuitry that is intended for child rear, which might be the reason why people are so emotionally attached to them. He also mentions that dogs even shed emotional tears, which the researchers have already tried to replicate. However, the attempt failed, and Dr. Sobel explains that that's just how science works, and that pure replication is rare in studies. There are also instances where replication fails, and this happened in a study concerning the effect of oxytocin on behavioral change in women. Dr. Sobel offered to replicate the experiment together with the lead author, but they refused, and they went ahead with publishing the failed replication in a journal. Dr. Sobel later published a rebuttal, which Daniel Kahneman advised him not to do because people don't usually read the details of a rebuttal, and it might cause confusion. Dr. Huberman appreciates Dr. Sobel's courage in publishing the rebuttal, and they both agree on the importance of replication and rigorous scientific methods in conducting studies. Dr. Huberman and Dr. Sobel discuss two factors that influence cultural perception of smell. Firstly, the human tendency to focus on outliers rather than similarities can obscure the fact that people generally have similar olfactory perceptions. To measure this similarity, researchers can use similarity matrices to determine whether people perceive smells similarly. This has been done for vision, audition, and olfaction, revealing that people's perceptions are most dissimilar in color vision, while their perceptions of olfaction and audition are similar. Secondly, unlike in other sensory systems, olfaction lacks anchors for language that would help people identify and describe smells consistently. It is difficult to determine if individuals perceive the same smell, as different people may associate an odor with different memories and experiences. Due to the rich sensory information and effective power of smells, scientists have proposed generating smells in silico through computer interfaces. This would allow individuals to experience certain smells without actually having to physically smell them. While this has not yet taken hold, it offers opportunities for expanding the potential of sensory experiences beyond what is currently understood. Google's recent startup, Osmo, is trying to digitize smell, as well as other companies in the field of research. Dr. Sobel explains that half of their lab is devoted to olfaction research, which includes developing a set of rules that link odor structure to odor perception. Until recently, there were no scientists or perfumers who could look at the structure of a novel molecular mixture and predict how it will smell or what molecular structure could or should be. However, within the past 2 years, a breakthrough has been made with rules being established thanks to the work of various labs. Dr. Sobel's lab also had a breakthrough with an algorithmic framework that can predict the perceptual similarity of any two molecular mixtures with high accuracy. They even generated olfactory metamers, which are mixtures completely non-overlapping in their molecular structure, but smell exactly the same, including a Chanel No. 5 mix. From there, the team can discover the set of components known as outer primaries that can be used to mix any odor that humans can perceive. The team's ultimate goal is to use a device to transmit smells over the internet. Dr. Sobel then goes into a proof of concept test conducted with Jonathan Williams from Max Planck in Munich, where they tried to recreate 4 different scents over IP. While they failed to recreate rose and anise, they were slightly better than chance at recreating gasoline. The team was successful in recreating violets, which marked the first ever scent ever transmitted over IP. Dr. Sobel admits that the concept is not yet practical, as the measuring device they use costs $1.5 million and is quite large. He believes that the device's size and cost will come down, but it will still take some time. When asked about the competition with Google, Dr. Sobel humbly admits that he would put his money on Google with their abundance of resources. However, he gives credit to small groups of creative people that may outgun and outrun larger groups. Dr. Andrew Huberman also brings up the potential implications of this technology for dating apps and other industries. They explain how olfactory digitization could facilitate medical diagnosis in the future as well. Dr. Sobel emphasizes that every disease is a specific metabolic process, which mean that it has metabolites that have a unique smell. Therefore, with the digitization of olfaction, it will enable medical diagnosis through an electronic nose in the bathroom for each individual. They envision that this technology is still far off, but it is an exciting prospect that various groups are working on. In closing, Dr. Huberman expresses his gratitude to Dr. Sobel and his team for their pioneering work and insightful discoveries in the field of olfaction.